All right. Good afternoon, everybody. Welcome to the afternoon session. This is Track 4, Science and Technology Innovations, sponsored by PRA Health Sciences. We hope you enjoyed lunch. My name is Kendall Davis, and I'm going to be your room host this afternoon, so I will be facilitating question and answers throughout the day as well. We are going to have the speakers do their presentations first, and we will have question and answer after they have spoken. If you're joining us via live stream, please feel free to submit your questions via the app, and I will make sure that we get any questions that come in via the mobile app answered by our panelists. And anyone here, if you are more comfortable submitting your question through the app, just go ahead and do so, and I'll make sure that they get responded to. So in today's session, Rare Research Resources, CRISPR Systems and iPS Cells, we will be hearing from professionals and advocates on these two technologies, approaches, and benefits in furthering rare disease research and the potential toward new therapies. During this session, we will be hearing from Marcelo Coba, Assistant Professor of Psychiatry at the Zilkha Neurogenetic Institute at Keck School of Medicine at the University of Southern California, and Lisa Flint, the Founder and Managing Director at the AADC Research Trust. So please welcome me and join me in welcoming them. Awesome. And our first speaker is going to be Marcelo, so please take it away.
So this is...
Yeah.
Thank you for the introduction. Thank you for coming, and the organizers for inviting me to give this talk. And basically what I want you to get after this talk is that there's a lot of discussion about using pluripotent stem cells and the uses. And I want you to have a better idea what things can we do with things we are not getting there, and how we combine different models for stem cells, animal models, how we do genetic engineering on these models to modify to get what we want to get. So what is a pluripotent stem cell? I think this is, I knew this was going to be useless. So during cell development, you have cells that you will have the capacity to generate almost every cell type or every kind of cell type. And that's what we will call a pluripotent stem cell. So it's a cell that has the capacity to generate almost any other cell type in your body. So if you look at there in green, I hate this. But in green here is what you were going to have as a pluripotent stem cell. These are cells that can renew, so they can proliferate, and this is important for something I'm going to show at the end. So they can they proliferate, you can culture them, and you get a bunch of them. They are dividing cells, and they start to differentiate. When they start to differentiate, they are becoming something else. When they become something else, they start losing the capacity to be pluripotent. It's like if you are having decisions in life, and you take one road, one way, maybe you cannot get the other one. So each time that the cell gets a decision, they cannot do everything and it can do when it was a pluripotent stem cell. So if you can see in that picture, from that pluripotent stem cell, you can cells from different lineages, and at the end, so in the middle panel, they still have some pluripotency. For example, a cell one of these cell lines can still generate every single type of cell that exists in the brain, and then when they become different cells or different brain, they cannot, they are just fully differentiated, so they are not pluripotent anymore. But from years, we know that even these cells that are fully differentiated, they still keep this capacity to be pluripotent. So this is what we are using now, what is our interest in just fully differentiated cells. We need to get these fully differentiated cells back into a stem cell. So that's what all these processes we are doing. So if you see, I think it should, yes. There in the circle, we have a fully differentiated cell, and what we're trying to do is get back to an iPS or a stem cell. So I'm not going to show how we do these things, but you will need to play with lots of different factors, some external factors, some intrinsic factors. So you need to stop the cell being the differentiated cell and doing what it's usually doing. And then it are what we call transcription factors, where we modified what genes are being expressed in these cells. And at the end of the road, which it takes some time, you get a stem cell back from a fully differentiated cell. So, but this is an important slide, and this is what things can we do, or what are the most common uses of these stem cells. And this is two main things. You might try to do cell therapy with them or disease modeling. And what you get here is, for example, we can start in the middle with a patient with a particular mutation, a rare gene mutation. In this case, it might be a gene expressed in the brain. We work with diseases mostly related with brain diseases. So what you get is from this patient, you will need to get a sample, which in general might be the two main things that you usually get are blood sample or fibroblasts from skin biopsies. From then you transform those cells into pluripotent stem cells. So you have these cells that carry the mutation that has the patient. And then you need to correct those cells for both cases, if you're going to do a disease modeling or if you're going to do cell therapy or try to do these two things. And I'm going to show later what methods are we using in doing that. But we correct the mutation. After correcting the mutation, you need to differentiate your stem cells that are now corrected into the cell type that you are interested. In this case, are neurons. And the idea of cell therapy is just to get those corrected in this case, neurons, back to the patient. But it's important to know that we are not there yet. I'll show you some examples, but what we are able to do this at best is in animal models like rodents. The other important path is doing disease modeling. What it means, disease modeling? Again, we get the pluripotent stem cell with the particular mutation. We correct it. And this is important to understand why we corrected those cells. So we work and model the disease with the cells from the patient, and we correct it to have a proper control. So we have a control with the same genetic background. And this is very important to do. It's not always done, and it's difficult, but that's, at least in my view, the correct way to go. You have the cell line, you correct it, and that's your control. And then you can do many, many different things to model the disease. I'm going to show examples of those. But the idea, for example, here in this picture is that, okay, you can screen. So you will have from these cells, they will have a particular phenotype. For example, just if these are neurons, and you have a mutation that impairs the function of these neurons, somehow the electrical activity. You may have an array that measures that electrical activity, and you see that these are abnormal when you're comparing with the control. So you have a battery of drugs that are trying to restore that function in the neuron. And that's how you start modeling on what is happening with the disease, and then you come up with those treatments to the patient. So, as I said, cell therapy, we are not there. The closest thing that has been done is sickle cell anemia, because this is a hemoglobin gene that has been corrected. For our view, that's something simple to do, or at least simpler than just trying to get the cells into the brain. But this is something that has been done. Challenges, as always, these are safety conditions, the safety concerns of what are those cells. So we are good in restoring activity of a particular gene or restoring the mutation, but then the cells that you get, there's still safety in going back to a human. And it's important with this is that not every disorder is going to be able to be, if you want, treated in this way. That will depend on what type of mutation, what type of disorder you have, what type of organ are you targeting. There are lots of different things. But in any of those, there are still big safety conditions. And one main thing for those is cancer-related conditions, because you are going back with cells that are still proliferating. But there's a long way there on that regard. So the other thing what I was saying is, genetic manipulation, genetic engineering. So this can be complicated. The CRISPR, it means cluster regular interspatial palindromic repeats, which is not telling you much about the disorder, at least in a simple way. It still is telling you related what is it doing, but it's not so obvious. But the CRISPR, just to see what it is and how is it doing, this is a defense system for bacteria. So you need to think, is it a system that the bacteria sends an exchange pieces of DNA or RNA coming from a phage, a virus, or another bacteria, and then target that DNA that is strange to the cell and cut it. So find and cut is what it does, the CRISPR system. And we hijack that for genetic engineering with an extra bit of the differences. What we are going to do is just the same thing. We use it to find a region of DNA of interest. We cut the region of interest, and then we repair. So for us, if you want to say what is doing CRISPR in our lab, it's just find, cut, and repair. And this, probably you cannot see anything on that, but it's showing you basically how it's into it. You need a guide to tell your enzyme that is going to cut the DNA where to cut. So just for example, for you, you have a particular mutation in a sequence of DNA. You need a guide to target that sequence. When it targets that sequence, you are going to have the enzyme that it cuts, which is the CRISPR. Is going to be the Cas9, which is a kind of scissor. Now we have different enzymes that do that. The Cas9 was the first one and is the most used one, but we have now some other different tools that are nice to use. So the enzyme is going to cut that piece of DNA, and then we need to provide a sequence of DNA that is now the normal one. For example, you have a mutation. You cannot really see it there, but it shows a mutation. So you have a mutation in the base pair. You provide a piece of DNA with a correct base pair. Because what is going to happen, you need to understand, is that once you cut that piece of DNA, the cells are going to repair it. And we're going to repair it with whatever it has there. So it's going to repair it with a piece of DNA that you cut, with another piece of DNA, or the one that you provide. So it's going to put back something. When it puts back something that is the correct sequence, is what you say that you corrected. So obviously, if you think about that, there's a lot of playing with efficiency there, because it's going to put lots of different things back in different ways. So in a dish, this is not a problem. So that's what kind of is trying to show there. But what you're doing is that you have your iPSCs, you cut it, you do the repairing by CRISPR, and then you will have what we call clones, different cell lines that you are reproducing in a dish. Some of them are going to be correct, and many of them are not. What you need to do is sequencing and see which ones are correct. In a dish, that's not a problem. You just check. It's a problem in terms of timing or how you do it, but you're going to have, hopefully, some corrected clones. When you get them, you just isolate them, reproduce them, and you start working with those clones. What I mean about that's not a problem in a dish is that CRISPR might be something that people will be interested in. It's something that you will be interested in. It's something nice to think that you can correct a mutation in a patient. You need to think about many of the things. We are not there yet at all. There have been some animal models. I think it's just got some examples from Duchenne and cystic fibrosis. People have been trying. But again, there are lots of safety concerns when we are saying that we are cutting specifically here, and it's true, cuts specifically what we want, but it might be cut in some other places. And then it's also the efficiency problems. And so there are lots and lots of safety concerns, and we are not there for humans. But it's a nice thought that in the future you might use this or some related method to correct mutations. Other thing that I don't know if I will have time at the end to show some other applications, but in the same way that you can correct the mutation, you can do lots of different things to model a disease. Just for example, you can add transcription factors in the same way working with CRISPR just to increase or decrease the amount of the gene of interest. So it's expressed more or less protein. You can add fluorescent tags. So for example, your protein, you cannot really see the protein in a cell that you're interested to look at because the amount of the protein is very, very low, and this happens to us. So what you can do is by CRISPR insert a fluorescent tag, and then you can see the protein. The nicest thing with this is that you are working with what we call the endogenous gene. So it's the proper amount of the protein that is being expressed in the cell. So there are many different things that you can do with this method to model the disease. And when I say model the disease by CRISPR, is another thing that is really, really important. This is good, and you will need to know what can give you and what can't. And the way to model the disorder, any of these rare diseases or any disorders, so you will need multiple different models. We still use a lot of mouse models or animal models. You will need to do this. It will allow you to ask different questions, and you will get different answers for that. You can do iPSCs, human, and in our case, derived neurons, as I explained, or also what you can derive from the same way, doing CRISPR, but instead of culturing in, people used to call them 2D, which is that the cells lying flat in a dish. What are called 3D models or organoids. Obviously, they're all 3D. I don't know where they come with the 2D and 3Ds, but they are all what you call organoids, spheroids. They grow in a way and with a pattern. If you want, I'm going to put them all this like this, growing like a brain kind of a structure. It gives you a structure. That's the difference. The other difference, when you do the neurons in a dish, you can do different neuronal types. In the brain, you have tons of different types of neurons and other cell types. So by playing with what transcription factors and what methods are you doing, you can do an excitatory neuron, you can do an inhibitory neuron, you can do a serotoninergic neuron. When you do an organoid, you get them, hopefully, most of them or all of them. So you start asking different questions, mostly how is the structure or how you can see the layering of the brain. So these are really, really useful. Obviously, in all of them, you will measure electrical activity, because for us working on the brain, electrical activity in all of different forms is one of the main readouts. So just a brief thing, what is a neuron, or what are we looking in the neuron? We mostly look at synapses. Lots of these rare disorders that are somehow impacting the brain are proteins that are expressed at the synapses. Those things are the synapses, the things at the tip of a neuron. You have a cell body, which is at the middle with the nucleus, a long blue thing, which is the axon, which at the end is going to end up in the synaptic terminals that are going to release neurotransmitters, and they are going to impact or transduce a signal on the postsynaptic side of a synapse. So that's what is shown there, is how the signal is transmitted. So you have the depolarization coming to the synaptic terminal, and it's going to be slow, and you can see it. And then transduce the synaptic vesicle release to the synaptic cleft, and it's going to be sensed by the postsynaptic side of a synapse by receptors. So that part is the synapse. So you have the action potential there, synaptic vesicle release, and now it's going to show that. Those are synaptic vesicles full with neurotransmitters, and going to impact on neurotransmitter receptors at the postsynaptic side, and then the signal goes on. So lots of these molecules are localized at the postsynaptic side of the synapse. So we're going to be a brief example of one super rare disease is occurring in just a couple of families in Saudi Arabia with a particular molecule that is localized at the synapse, and how we were using with all these things. So this is a mutation in that kinase. It's a protein that has protein kinase activity, which means that it phosphorylates other proteins, but it doesn't really matter. It's an enzyme with a function. So they have a mutation, a homozygous mutation from both parents' alleles. And what it produces is a total knockout of the protein. You don't have the protein anymore. Those patients don't have the protein anymore. And it's a single mutation. So we repair it by CRISPR. We induce neurons, We got neurons, and what you get there is, if you have all those bands, what is measuring is proteins. You see what are those arrows there, and then you have 2 lanes with nothing there. Those 2 lanes are the patients, which is a single mutation in the DNA. We corrected by CRISPR, derived neurons, and then you can see the protein there. So in our hands, in a dish, this is really, these days, pretty simple to do. You can correct it, and you can revert the phenotypes of those mutations. But something, just very briefly, to end up on how you need different models to study diseases and what it can tell you when you're working with these things. I say that we were working this with the neurons. So neurons, they don't proliferate. They don't reproduce. When we were studying the cells, we got a period that you can with the stem cells that you proliferate them, and then what you get is neuronal progenitor cells. We started to look that these cells didn't proliferate well. This was not a phenotype at the synapse. Even if we were studying the synapse, we observed this in the cells. So we went to the animal model, having the same mutation and a knockout, and asked, okay, is this something relevant for the disease? Do they have something similar? And we look for a particular place at the brain when you have this type of cells that can reproduce, that can proliferate. And what we've seen in the animal model is the same as our cells in the dish. They have a decrease in proliferation. The next thing, I just finished. I want to show this thing. The next thing is, okay, we know that these cells that are able to proliferate are involved in a particular type of cognitive test, or it allows you to do some particular task to humans and to mice as well, which is the ability to discriminate patterns, to see something that is similar to other things. That you look at something and say, this is similar, this is not similar, which is called pattern separation. And the nicest thing with this is that you can do this with humans at the touch screen, and you can do the same test to mouse, and this is a mouse doing those tests. We can take the volume down. It's a test that mouse, what it needs to do is recognize different patterns. When there's that beep, he knows that needs to go to the screen and touch the correct answer. And he's doing that because then if he gets the right answer, he gets food. See, touch, go back. And I think that was the last one. So you can do the same as human. Human with this issue, that they will have they will perform badly in pattern separation. You can do the pattern separation using the same touch screens to the mouse, and the mouse also has a deficit in that task, as you were saying. And it was coming just for the observation that these cells were not proliferating, but we were looking something else. So obviously, this or an electrical activity of different things might be your endpoint. And then you can start to play on how to correct these deficits, in this case on behavior, or in particular, or electrical activity in each model. So probably I'm skipping this. This is just to show other features that we can do, like express a protein that you cannot really see by CRISPR. We put a fluorescent tag, and we now can start looking at that protein and what is it doing at different regions in the neuron. But the important thing with that, what I wanted to show you, is that how important it is to play with different models for the disorder, and for you to have an idea what things can be answered with one model or different models. And most importantly, for all of these disorders, any disorder really, when you have a mutation, you are focusing, for example, in our case, it was a synaptic protein is going to do something at the synapse. These are all developmental disorders. And they are going to do, the function of these proteins most likely are going to be different through development. And they are going to localize at different regions, in this case in the neuron, but it's applicable to other cell types. And they are going to have different functions. And it's important for you to know at what stage that or the phenotypes that you are getting for the disorders are due to what. It's an endpoint in a mature cell. It's something that is very early on as a proliferation cell. In our case, for neurons, it might be, if it's because it's localized at the synapse, it's because it's localized at the presynaptic site. Molecules have different functions through development, and depending on where are they localized. So it's not going to be only one thing, and you need to look at many, many different things. And with that, and that's where my lab is localized. Here is in LA, so it's just short transfer here, and some of the collaborators that we have on funding. Thank you for attention. Questions are at the end?
Yeah.
Or, okay.
Wonderful, thank you so much. And now, Lisa Flint.
So what's that one? Just the green one.
Now.
Hello, thank you for having me. My slides are nowhere near as technical as Marcelo's there, so I'm just going to brief you a little bit about my organisation, myself. Firstly, I want to thank Global Genes for putting on the summit and for all of you for being here. My name's Lisa Flint, and I'm the founder and director of the AADC Research Trust based in London. So a little bit about us. We were established in 2006, a UK registered charity. We support 70% of the world's AADC sufferers, which actually doesn't amount to much because there's only 130 to 150 of us anyway. When I say us, my son suffers with the condition, so I talk about us as a group because my son suffers with it. So we work closely with an international expert medical and scientific board. We've raised over £1 million for research. A biochemistry overview. So we're autosomal recessive metabolic neurotransmitter disorder. It disrupts and blocks the production of two of the most essential neurotransmitters, dopamine and serotonin, and it causes a deficiency of dopamine, serotonin, melatonin, and mostly cerebral folate. A superfluity of L-dopa, the precursor for dopamine, and 5-hydroxytryptophan, which is a precursor for serotonin. The symptoms are catastrophic. So as with a lot of the rare diseases and lots of the stories I've heard since being here are very, very similar across these diseases. Here you can see a list of the symptoms predominantly that affect most of the children to a greater and lesser degree. That's what we're learning about at the moment. So the geographical epidemiology, so 130 to 150 across 30 countries. At least 3 children die every year from this condition, but in this year we've already lost 6, so it's quite devastating. Some of our past research projects, so the PhD project, just in the basic understanding, when I set up the foundation, I always believed that we needed to start right back at the beginning. We first described the disease back in '89. By the time we set up the foundation in 2006, not a lot had happened. We still only knew of about 30 patients by that time. And we needed to really start back at the beginning. So we employed the help of a PhD to do that, to really start the building blocks. We've created a registry. We have a biomarker for newborn screening that's suitable for that. So we have an international database. We've developed a mouse model, and we standardized the AADC enzyme test in London as well. We also have current research projects, one being the induced pluripotent stem cell PhD project that we are now in our 3rd year, almost completed, which I'll talk about a little bit more in a minute. We have a genetic variation project looking at the genetics of our disease to see how that affects the disease outcome and the mild, moderate and severe spectrum. And a scaling as well, that's similar to Parkinson's disease, because actually our disease presents very similar to Parkinson's disease, and we share the same drugs as that particular group. We do have 2 brain-delivered gene therapy projects. One of them was originally in Taiwan, started back in 2009. 24 children have already received that brain-delivered gene therapy, and that's had quite a significant outcome on the disease, and actually that's been developed into a drug as we speak, as PTC are involved with that now. The US, which is an alternative brain-delivered gene therapy, is going to a different part of the brain. 6 children have now taken part in that first phase so far. So we're excited about the next couple of years and having comparable data about the delivery of these 2 gene therapies to 2 different parts of the brain. So on to the successful creation of induced pluripotent stem cell lines for 2 of our patients suffering aromatic amino acid decarboxylase deficiency. I want to thank the Caroline Kramer, who actually is our PhD student, been working on this project for the last 3 years. And she presented a poster presentation and published that. So she, like I said, she's just about to finish this project. Here is a few of her slides. So you can see that she's taken the skin fibroblast from those 2 patients and she's converted those into dopaminergic neurons. So one's a homozygous mutation, the other one's a compound heterozygote. So we have 2 different variations of these stem cells. Here's the staining of the neurons. You can see the top line is a control line, and then we have patient 1 and patient 2, and you can see how that differs from that. Here we can see the enzyme activity from patient 1 and patient 2. You can see a reduction of enzyme activity compared to the control patient that's at the normal level of that. I'm not really sure how to interpret the protein, to be perfectly honest with you, because I don't know why patient 1 would show a greater production of protein and patient 2 shows a reduction, and the control actually is in the middle there. So actually, I don't know how to tell you the explanation of that. Maybe Marcelo go. So that's the end of those slides to do with that poster presentation. So just to thank Caroline Kramer for that and that brilliant work, which has now produced us 2 cell lines that we can work with. So what can we learn from these? What, as an organisation, a patient advocate, do we want to know about these cell lines? I'm going to use my son as an example. He's 20 now. And he takes a myriad of medication. Here you can see most of these are used in Parkinson's. Half of them are used to deal with the side effects of the other half of the drugs. So we're not even treating the condition anymore, we're treating the side effects of the drugs. So we want to know more about what's the longevity of Jake's disease and how these medications are going to affect him long term. Jake, I must say, of the 150 patients, is amongst 5% that actually respond to medication. So most of them have no mobility, no speech, and catastrophic outcome. We are one of the lucky ones within our population because he does respond to the medications, and we have a greatly improved quality of life as a result of that. So what I want to know as a family with a son that has a rare condition is what I can do with these induced pluripotent stem cell lines. So I want to know if I add all of Jake's medications to these and we stress these cell lines, what's that going to do to that cell line? What's the outcome for us? What are we looking forward to as we do this? Can we withdraw some of the medications? Can we look at what the reaction is to this cell line once we withdraw some of the medications? Can we actually add untried medications? Most of our medications address the dopamine pathway, whereas the serotonin pathway is largely left untreated. So can we add some serotonin beneficial drugs to this? Will it have negative effect against the dopamine drugs because you wouldn't normally put the 2 together? What other important information can we find out about this? So translational medicine is an exciting prospect for many of us suffering rare diseases. Can we test some of these pioneering breakthroughs on these lines? They may be a one-time only treatment with catastrophic or irreversible effects, as well as the potential for providing life-changing benefits. So to trial gene therapy, CRISPR, the chaperones, enzyme replacement in these cell lines would actually be hugely beneficial. It's all knowledge. Other information that is useful to us, if we create the iPS lines before and after trialling medicines and therapies of any sort, we can monitor biochemical and genetic effects that these translational breakthroughs may have on our community, and we can measure those against the clinical presentation. So sufferers of rare diseases rely heavily on drugs usually developed for entirely different diseases. So reaction and responses to the drugs using iPSCs would be great. They're highly speculative and unpredictable for AADC deficiency, so these lines would be very, very important to us to find out more about our disease. Some medications could have dire consequences as well as life-changing benefits. So a question. is individualizing treatment strategies by trialing medications, exploring genetically engineered breakthroughs, using your own induced pluripotent stem cell lines first, the way forward for those suffering rare diseases. Unfortunately, we can still only hypothesize the answer to this. That is until we discover the answer, and we will always discover the answer. I'd like to dedicate this presentation to every child who's lost their battle with AADC deficiency. Most of those I've known personally over the 13 years that I've been working with this disease, before we actually have a chance and had a chance to save them. So this goes out to them. I'd also like to thank my co-speaker here, Professor Marcelo Coba, for his presentation, and to all of you for listening, and we hope you enjoyed it. Thank you to Nicole Boyce for this amazing summit, my first, hopefully not last, for her personal words of wisdom, and she'll know what that means, and to all the Global Genes team for putting this event together. So please enjoy the rest of your day.
Wonderful, thank you. I will walk around, so if you have questions, just raise your hand. We do have about 20 minutes for question and answer, so just raise your hand and I'll bring a microphone over.
Marcelo, wondering about CRISPR delivery and how that's done in a petri dish versus a human. What's the vehicle to get CRISPR into a cell?
Now, what we haven't been trying in humans is mice, and there's a lot of questioning. These things that we don't know. CRISPR is very recent; it's just 5-6 years old, and honestly, for treating patients, we don't know what might be happening. The first thing, even if you get it in the liver, the first thing is, as always, well, is how treatable is your disorder. But the first thing it will be with that is, what we say is specific when we are cutting is, okay, that's a specific, and there's a way that you always say, how many non-specific targets are you going to have, and what are the off targets? And there's a very recent publication a month ago that they have found tons of weird things, cutting big chunks of DNA. For our work, in a dish, it works. I've never seen these things that have been described in mice. But there's a way to go to apply it to animal models and longer to humans. We got to think that you need to target a piece of DNA with a guide. Think that that's super specific, and that's the only thing that it's doing, going there. And it goes just by base pair. So it recognizes a sequence of DNA. The first thing that you have two genes that are very similar, for example. There are cases that are very similar that the sequence is almost the same that you will need to target. For sure, it's going to target the other one. There's no question about it. There are lots of these things. It will depend, and we're not, and these are things that we know. There are other things that you will need to do really heavy, in-depth sequencing to every time that you're doing, and see how you keep the lines, and when we are still reproducing them and keeping what is really happening in the long term that we still don't know because of it. This is very recent. We are still learning. For disease modeling, it's working great. And this is the first thing that we are focusing. The other, it will be coming. And obviously, there will be lots of regulations to start using any animal model first. But it will take some time.
I just want to add a comment maybe to Dr. Coba here with regards to delivery of CRISPR systems to humans. Been looking at this issue, so just to say where things are standing right now, there's some work. I mean, most of the work is done with what's called the RNPs, which are a ribonucleoprotein, which is a complex that is made outside of the patient from the Cas9 CRISPR protein with the guide RNA. And this can be administered as such, but to a limited amount of systems. For example, the eye by injection can be targeted, and hematopoietic stem cells can be targeted this way outside of the patient. But for systemic delivery, there's also work using viral vectors such as AAVs and lentiviral vectors for delivery of the entire CRISPR machinery. So as Dr. Coba said, this is all very preliminary work, and there's a lot of hurdles in the delivery of the CRISPR system, which is similar to siRNA therapeutics or other genome editing modalities. So the delivery is definitely one of the major issues that we have to face.
Thank you. I do have a question from our online audience. So one issue I have heard about specifically regarding neurons with iPSC cells is that they stay immature and don't turn into normal somatic cells. Is this an issue still, and how does it affect modeling and treatments?
Yeah, that's totally true. All systems that are working with iPS cells, what you get is an immature cell. We're working with neurons, we get immature neurons. What does it mean to be immature neuron? I will just say that this is not the fully mature neuron that we used to have when we work with a mouse model. How that affects is, well, knowing it's just knowing what you have and what are the questions that you can present to the system. These are developmental disorders, so they are presenting you with, if you know that you are modeling an early developmental phase of the disease, it's good with you. For example, in our case, it's our cells. Do they develop nice, fully mature synapses? No, but they have synapses that we can map and match to what is particular stage in the mouse. We can work with them, and we know what we are getting. But it's true that you don't have a fully mature. That's what I was saying, what you need to play with different models. That each one, it will give you something different.
I had a question on the left, over here. This is for Lisa Moore. You had mentioned in your talk there's a clinical trial right now of a few people undergoing gene therapy in another country. And Marcelo mentioned that there's a lot of regulatory hurdles here in the US. Is there a place in the world where there are less hurdles that people are sort of heading towards for more clinical trial treatments?
The original clinical trial for AADC deficiency happened in Taiwan because there was the biggest single population of AADC sufferers there at the time. And we were very fortunate that the original gene therapy was designed for Parkinson's disease. And unbeknown to the scientists developing this particular technique, the enzyme that they discovered that was reducing in Parkinson's disease was the therapy they created as a result of that was to replace the AADC enzyme, which is the enzyme that's missing in our group. So that was originally delivered to a monkey model in Japan many years ago, and it continued to express dopamine for about 7 years, and it was then that it was modified to suit a group of AADC deficiency children in Taiwan, and that's where the trial, the 4 phases actually took place. The license, I believe, for that particular trial was bought up by a biotherapeutics company here in the States and subsequently PTC just a couple of months ago. So they are now taking on the responsibility for taking that to market as an actual available drug, probably first here in the US and then to the rest of the world, hopefully. The second trial was equally a Parkinson's expert scientist working on AADC for Parkinson's and modified again. But it was his belief that delivery to the substantia nigra versus the putamen that was in Taiwan would give greater benefit to our sufferers. And we've yet to see the data from that. That's in its early stages. So as I say, 6 children have received that particular gene therapy. The regulatory processes for that trial here in the States was extensive and has been going on since 2011, 2010. We funded actually the regulatory administrator to write up the extensive paperwork that needed to be submitted for that trial. So we've driven really both of these gene therapy trials, and you know, where there is a will, there is a way. You have to maybe shop around and don't stay local with your science. Go to the world. There are some brilliant people in lots of countries. And I work with a collaborative group from 15, 20 countries. They're all doing different aspects of AADC. So, I've always felt that the building blocks of this disease comes from the very, very beginning. So we have a patient registry. As I said earlier, we have a natural history study that will be coming through very, very soon. So all of the background work has still been going on all of the time. The gene therapy regulatory processes has been taking place. It's frustratingly slow, even from one phase to a second phase. Here in America, it is frustratingly slow. We don't have that time. We lose 3 children every year. We've lost 6 this year. 4 of them are Americans. So we don't have the time, even when listening to Marcelo talking about CRISPR. I spoke to him before we had this session because I now want him to take on the responsibility of our iPSC lines and see by performing CRISPR into those cell lines what happens, have a look at them, and what we can do with them. It's, 10 years maybe down the line, but for those children that are surviving 10 years on a genetic repair, fantastic. If you repair the gene and you've kept them in the best possible health for all of that time, then, we're just 10 years down the line. Actually, in, those particular terms, it can move quite quickly. So, you have to start somewhere. I'm hoping that now I've announced that publicly that he's going to feel the responsibility to take that research on. So, because it's live streamed as well. So, but yeah, so, anybody embarking on gene therapy, using the vectors. What we have done with AADC deficiency that is going to benefit single gene defects is shown that the safety data, so forget about the outcome, the safety data through brain delivery gene therapy is doable. For us, the outcome has also been beneficial as well. And I think Canavan's disease was one of the first diseases to do the brain-delivered gene therapy, but I don't think there was particularly a great outcome for that particular population, unlike AADC deficiency. So we've been very lucky. We've been lucky that we've ridden on the back of Parkinson's disease research. We've been lucky with our group of scientists. We're a very intimate group. We don't go out publicly very much to share lots of what we've done, and I haven't gone out there to massively raise awareness about the disease because I don't have anything to offer families that do come forward at the moment. Well, we do now. We didn't 5 years ago. So now we have established a base, a very well-established base. We'll go out and try and find those patients, and hopefully the gene therapy, CRISPR, the induced pluripotent stem cell lines will help us understand the disease. I still strongly believe that every child that has AADC deficiency should have a cell line because the drugs that we use for Parkinson's don't work for the majority of our group. So we need to try them in the cell lines because they do have catastrophic side effects in our group. So it would be better to be able to personalise treatment that's available today and look forward to the genetic engineered treatments that are going to be available in the near future and still have the lines available to try them there first.
Thank you both for coming today. I'm over here in the corner. I know everybody. My question, I'm finishing up Jennifer Doudna's book, A Cracking Creation, so I highly recommend it to everybody here if you're trying to figure out about all the CRISPR stuff and what's going on. But as I read through it, there's in vivo and ex vivo options for editing. My thought, and others have shared the concern, and you did as well, about these off-target mutations that are occurring when CRISPR goes in and cuts things that aren't being targeted. Ex vivo can, I know this is cutting edge, but can you then, what I understand is outside the cell you do the CRISPR, or outside the body you do the CRISPR editing, and then you reproduce those cells and place it back into the body, and along the way you're checking theoretically for the off-target editing that is the fear. Is that?
That's more simple in terms of off-targeting, and delivery is a different thing. And yeah, it has their pros and cons. You can check and you know what you did with that genome. And you can select very well. But then it's not everything is going to be solved, but getting back then. We're working mostly in brain, and in brain you also, you will need to think that the cells need to go to the proper place, to the migration that already occur, and how it's going to do it. There's people that are super skeptical and that you will ever get that. It will depend on what disease are you treating. There will be things that can be done, and maybe in other organs or systems you can't. But it's true that in terms of off-targeting and delivering, well, you are not delivering all the viruses, and it's different. But you have other problems. And also, as I say in the talk, the safety concerns are different. In this case, is that if these cells are going to start to reproduce, and they get something like a hyperproliferation phenomenon similar to cancer. So there are other issues.
I have another one from our online audience. How might iPSC therapies be used to regenerate muscle tissue lost due to advanced muscular dystrophy? And what are the major obstacles that we are currently facing?
Yeah, that's happening, and something similar with Duchenne that have been trying in mouse models, and the reports that it worked to a certain extent. Usually, sometimes I have, not with that particular manuscript, but in general, manuscripts are showing you some of the results of what they've done, and you don't know how safe it's going to be, or it's true that worked, and there's at least one publication that they did it for Duchenne and working well. But, I cannot comment on much more on that. It's an animal, it's a mouse model.
Hi, I'm in the middle here. My question is about gene size limitations. I remember hearing about a gene therapy that used to replace a CEP290 gene, which was like a pretty small gene. It was like 7 or 8 kilobases long, and so they could insert it into the virus to put it in the body. And my daughter's got a condition where the gene is much physically larger. It's like 120, 140 kilobases, something like that. And I was under the impression that they could not insert that into an AAV vector because it's too big. That'd be my impression. But I don't know if that's right, and I don't know if CRISPR, changes
but
that.
CRISPR, it's different, but it is the whole package that you will need to insert, and with how many different viruses you will need to use. But in terms of gene, it's different what you are targeting. In this case, you need to provide with all this, the CRISPR machinery, but then it's working on your genome, basically. So it's not like you are replacing a gene. You are just correcting, for example, in this case, a point mutation. But it's true that, you need to get the whole package of all the machinery. But it's different. So it's not gene limiting. It's limiting in the way it's the delivery problems on all the machinery.
Wonderful. Do we have any more questions? Anybody else? All right. Well, join me in saying thank you again to our wonderful speakers. And I would just like to encourage everyone to go into the mobile app and rate the session. Let us know what you liked, and how you felt about it. And have a wonderful rest of your afternoon.